Alzheimer's disease is the leading cause of dementia worldwide. In the US, it affects over 5.6 million Americans. The greatest risk factor is age. So as we all age, we do increase the risk of developing Alzheimer's disease. And at the age of 65 and older, you have around the age of 65, there's a 1 in 10 chance you will develop Alzheimer's disease, and that risk continues to increase the older one gets.
Many of you might have experienced Alzheimer's through family members who have had Alzheimer's or might have it now, in which case you've seen the changes in behavior, you've seen the changes in their lives. Well, these mice, who are now healthy mice, once they have Alzheimer's, we'll notice the dramatic behavioral changes in them as well. Tell me a little bit about mice and why mice and why they're beneficial.
So mice are, of all of the model organisms that can be used, mice have a lot of benefits. They are mammals. They are relatively short-lived, and there's a lot of genetics in the mice that have already been investigated and different models of Alzheimer's disease that can be used. So we can model different aspects of Alzheimer's disease in different mouse models. After we dissect out the brain, we freeze half of it, and with that half that's frozen, we can look at molecular information, so gene expression, protein expression levels, and those sorts of things. With the other half of the brain, we fix it in, it's like formaldehyde.
Okay.
And so this is half of a mouse's brain, and then after it's fixed, we section it on, we can use a lot of different types of instruments to section the brain into very, very thin sections. And with those thin sections, then we can stain them and look for different components that have changed in the brain. And so in this plate, we actually have sectioned brain, but in each one of these wells is a different section of the mouse's brain. And so then with these sections, we can mount them onto microscope slides and do what's called histology. So we stain the brains for different proteins and cell types. These are MRI images from our mice, and the first one we have is a healthy control mouse. And really what I want you to focus on is this area right here. These are the ventricles, and they're very small. In this particular image, they show up as white. The fluid shows up as white, and you can see they're very, very small. Now if we look at the Alzheimer's disease mouse, which is the one here in the middle, you can see these white areas are very large. So that is corresponding to the increase in ventricle volume. All of that is fluid. And also you can see just the size of this brain compared to this brain. It's much smaller. So these are the Alzheimer's mice that were given the placebo, or the sugar pill. These are the Alzheimer's mice that, instead of getting the placebo, got the drug treatment. So they still do have larger ventricles. These mice were already sick. So in this study, we didn't necessarily expect to regrow all of the damage that had been done, but we were interested in whether or not we could stop the progression of disease getting any further. So what we saw is that these mice had much smaller white areas, and on this particular side it's a lot smaller, and the brains are actually bigger as well when compared to the mice that got the placebo. There are treatments that can modify behaviors, modify some of the symptoms, but actually preventing the disease, stopping the disease, or curing the disease, none of those treatment options are available. So when my grandmother passed away, I had just graduated college, and I decided that this was a very important question that I wanted to spend my life investigating. So we're really excited about this research, and it's very important because we are here in San Antonio for the first time showed that this drug combination has benefit in Alzheimer's disease. And not only does this drug combination benefit Alzheimer's disease in our model, but it has been shown to benefit so many other health conditions that occur with aging. And Alzheimer's disease patients, they have more comorbid medical conditions than other patients. So if all goes well, and this is effective at treating the brain, it may also help a lot of their other systemic issues that are failing as well to increase a healthy lifespan.